Hi, everyone. Thanks for tuning in. Um, so I'm going to talk a little bit about my research with Professor Ko. Um, so we were doing eye tracking research. We started last summer. We did DSSI together, and we worked with a local high school student named Adric. Um, and on the right, you can see a word puzzle displayed on the screen. It's called an anagram. And basically, it's the letters of a word, but they're shuffled randomly, and you try to figure out what word it is. So for this, the word here is a flavor. And basically, as you're solving it, looking from letter to letter, we track, which letter you're looking at with the eye tracker. So what we wanted to do specifically was get the sequence of letters people look at. So we wanted to get like this kind of data. Um, and it turned out that you couldn't actually get that data if you just had black letters on a white background. And the reason is there would be a lot of ambiguous fixations a lot of times where you weren't sure what letter people were looking at. So for example, here, you don't know what letter, whether it's T or I, that they're looking at. And so the way we were able to get rid of that was making the letters very difficult to see with peripheral vision, because presumably that person is looking at T or I with their peripheral vision. So we reduced the contrast between the background and the letters and made the letters much smaller and farther apart. And all those changes made possible the collection of letter sequence data. And so we ran an experiment in the fall, and we got 29 participants from the Drew Psych 101 classes. And we did data analysis. I had to write a lot of Python code to do that. And we were fortunate enough to be able to present this at the neuroscience conference at the University of Scranton. So I'll just kind of walk you a little bit through the results that we got. So one thing you notice is that in the letter sequence data, the first and last bigrams, like the first two letters and the last two letters of the solution word, tend to be located towards the end. And you might wonder why that is. And we thought it was because, if the participant read the first two letters or the last two letters, that would kind of give away the word. And so those were important pieces of partial solution knowledge that kind of developed gradually towards a solution. And so here you can see the location of the bigrams in the solution word. And for solved trials in blue, they tend to be located towards the end of the first bigram, also with the last bigram, but not with the second and third. So the second and third middle bigrams don't seem to provide much information about the word, the solution word. Another thing you can see is that the probability of looking at the first and last bigrams increases dramatically across the letter sequence data and becomes significantly higher for solved trials than for unsolved trials around the fourth to fifth quintile. Same thing for the last bigram of the solution word. So this was a nice result. And one thing we're going to try to do is I'm actually graduating, but we're going to probably run experiments in the fall. We've recruited some Drew psychology students who are going to be working with us. And basically what we're going to try to get is get data kind of like this, where the participant has a decent chance of solving the word and produces a letter sequence of a nice length. And we're going to be testing some other hypotheses, namely if there's a really common bigram in the English language, like TH or CH or something, will the participant immediately jump to that as hypothesis? So thanks for your attention, and let me know if you have any questions.